Hello students, I am your educator Dr. Hina Devnani and today we are going to study chapter number 10 of 11th standard. The name of the chapter is Cell Cycle and Cell Division. This video will deal with the first part of this chapter in which we are going to study about mitosis. But before studying mitosis, let us understand what is cell cycle. As you can see, the definition is given here. Cell cycle is defined as the series of changes that occur in a cell which leads to division of cells into two daughter cells. We have studied in first chapter that living organisms grow. Now how do they grow? They grow by dividing their cells. If there is one cell, it has to divide and form two cells. Two cells will further divide to form two more cells. That means total four cells. So in this way, they'll continue division of cells, and because of division of cells, the body of the organism will grow. So for growth, cell division is very important. And when this division occur in a cyclic way, there are series of changes that occur in a cyclic way, which leads to division of cells. So when these cyclic changes occur to divide the cells and form new cells, the whole process is called a cell cycle. And as we know that growth is very important, so we can say cell cycle and cell division are also very important processes that occur in a living organism. Now, this entire cell cycle is actually divided into two important phases. First is the interphase, and second is mitosis. Interphase is the common phase that occur in all the cells. Mitosis occur only in somatic cell. What are somatic cells? The cells apart from the gametes or the cells apart from the reproductive cells are called as somatic cells. Mitosis occurs in somatic cell. What happens in reproductive cells? Another type of cell division which is called as meiosis that we are going to study in next video. So here we'll be studying with mitosis. Now as I said, interphase is the common thing in both type of cell divisions. The average duration of the entire cell cycle in a human cell is 24 hours. That means human cell requires 24 hours to divide. Now out of these 24 hours, mitosis occurs only for 1 hour, whereas interphase occurs for 23 hours. Now what is the difference between these two phases? Interphase can also be called as the preparatory phase for cell division, and mitosis is the actual division of cell. So here the division takes place, whereas here the preparation for division takes place. And because this occurs for 23 hours, we can say that majority of the cell cycle is spent in interphase. And for mitosis, there is only 1 hour. This is the diagram of the entire cell cycle. You can see this is the mitosis, and from here up to here, this entire thing is interphase. So you can see interphase is very long phase. That's why it takes 23 hours to complete. Now let us see this. What is this? The first phase of interphase is G1. Second phase is the S phase. The third phase is the G2 phase. So these three are the sub phases of interphase. You can see here that here there is one cell. Here also there is one cell. Again here we have one cell. Here also we have one cell. But in mitosis what happens? One cell is divided into two cells. So actual division takes place in mitosis, whereas the preparation for this actual division occurs in interphase. Now one by one we are going to study all these phases. First, let us begin with interphase. It is a phase in which different changes occur to prepare the cell for cell division. As I said earlier, that this is the preparatory phase. So we can say that the changes that are required for cell division, the preparation that is required for cell division occur in interphase. Now this interphase is further divided into following three subphases. First phase is the G1 phase. G1 phase is also called as first gap phase. What happens in G1 phase? Biosynthetic activities occur at a very fast rate. Cell synthesizes proteins. Cell increases the number of mitochondria and ribosome. Now, why the cell is doing all these activities? The cell is actually preparing itself for DNA replication. Now, here what happens? Now, we have seen that human cell consists of 46 chromosomes, right? So, when a cell divides, it forms two different cells. Now, both these cells should have 46 chromosomes each. But a cell cannot simply double its number of chromosomes. So what a cell does? A cell actually doubles its DNA. You need to remember this. The number of chromosomes is never doubled. What is doubled? Amount of DNA is doubled. That means DNA replication takes place. You can see in this diagram. See here, these are the chromosomes. Here you can see. Now here it became double stranded. You can see here 2 strands are there, chromosomes, right? So the number is, here the number of chromosome is 2, here also the number of chromosome is 2. Only the amount of DNA has increased. The number of chromosomes never get doubled, the amount of DNA gets doubled. And the DNA is doubled by which process? By DNA replication. Entire process of DNA replication will be taught to you in 12th standard. For now, you just have to remember that DNA replication is just doubling the amount of DNA or making copies of DNA. And these copies are made in which phase? S phase. So now for making new DNA, obviously copies are being made, that means new DNA is being made. The cell requires a lot of raw material. And from where does this cell get raw material? It gets from the cell itself. So the cell has to synthesize this raw material first before the DNA gets replicated. That's why we say that G1 phase is actually the preparatory phase for this S phase. Don't get confused here. S phase is the phase where DNA is replicated. That means DNA is doubled. G1 phase is the preparatory phase in which all the raw material is formed. All the material that is required to form new DNA is prepared in G1 phase. So G1 phase helps S phase. If there was no G1 phase, S phase could not be occurred. So we can say S is the phase where DNA is replicated. At the end of DNA replication, each chromosome is with 2 sister chromatids. You have seen here, these 2 chromosomes have single chromatids. These 2 chromosomes have double chromatids, 2 chromatids each. So the number of chromosomes have not increased. That means the ploidy is not increased. The ploidy remains same. What has increased? Amount of DNA has increased. So we can say that the amount of DNA gets doubled during the S phase. Now we have understood these 2 phases. Now let us understand what is G2. G1 is first gap phase, G2 is the second gap phase. G1 can also be called as growth phase, first growth phase, and G2 can also be called as second growth phase. What happens in G2? Here the cell will prepare itself for mitosis. When you see this diagram, you can understand. First there is G1, after G1 S will be there. So we can say G1 is the preparatory phase for S. Now there is G2, after G2 mitosis is there. So we can say G2 is the preparatory phase for mitosis. So here the cell will prepare itself for mitosis. It also involves protein synthesis and further growth of cell. Let us again look at the diagram. What is written here? In G1 phase, what happens? Cellular contents excluding the chromosomes are duplicated. Everything else will be duplicated, but not the chromosomes. In S phase, what will happen? Each of the 46 chromosomes is duplicated by the cell. Chromosomes will be not the number, but the chromosomes will be duplicated. That means it will have 2 strands or 2 sister chromatids. What happens in G2? The cell double checks the duplicated chromosomes for error, making any needed repairs. If there is any problem in duplication of DNA or in these sister chromatids, in G2 phase, there will be checking of these problems and there will be repair. And then there is M phase, that is a mitosis phase. Let us start with mitosis. It is also called as equational division. Why? Because the number of chromosomes in the daughter cell and the parental cell remains the same. This I said earlier, number of chromosomes will always remain the same. In mitosis, in meiosis, it is reduced. That we'll see afterwards. But in mitosis, it will always remain same. That's why mitosis is also called as equational division. Now, how this mitosis starts? It starts with nuclear division. That means initially the nucleus will divide. So there will be one cell, there will be one nucleus. This one nucleus will divide and form 2 nuclei. After this division of nucleus, the cytoplasm will divide and the entire cell will divide. So when the nucleus is dividing, it is called as karyokinesis, and when the cell itself is getting divided, it is called as cytokinesis. What happens in this nuclear division? Chromosomes get distributed equally during mitosis. We have seen that before this mitosis, in S phase, chromosomes get sister chromatids. That means each chromosome is with two sister chromatids. So here in this division, what will happen? In both the cells, one sister chromatid will be distributed. So the number will remain same, but the chromosomes will again be with single chromatid. Mitosis takes place only in somatic cells. Somatic cells are the cells apart from the reproductive cells. Mitosis is further divided into following four phases: prophase, metaphase, anaphase, and telophase. Let us see one by one. First, we'll see it through images. Let me explain you this thing. This is a cell which is in interphase. That means still it has not entered the mitosis. Here you can see some thread-like structures. These are actually the chromosomes, but they are not in condensed form. So we can say that they are chromatin network. They are in the form of thread-like network, which is called as chromatin. When the cell enters mitosis, slowly the condensation of these chromatin fibers start, and they look like this. These are the chromosomes, the condensed chromosomes. This is the diagrammatic view, and here you can see these photographs have been taken from microscopes. This is the metaphase. In metaphase, what happens? All the chromosomes will be aligned in a single plane. You can see here also a single line, and here also they are aligned in a single line. All the chromosomes will be arranged like this. This line or the equatorial area is called as metaphasic plate. It can also be called as equatorial plate. In anaphase, what will happen? These sister chromatids will get split from the center, and this one chromosome will be divided into two parts. One chromatid will go here, the sister chromatid will go in other side. This phase is called as anaphase. Here also you can see it clearly, and here also you can see. The chromosomes are being stretched and they are moving towards opposite poles. After their movement, what will happen? Telophase. Here you can see this is the nuclear membrane. In prophase, the nuclear membrane will start to disintegrate. Here there is no nuclear membrane, here also there is no nuclear membrane. But in telophase, the nuclear membrane will reappear, and you can see that there is one cell with two nuclei. So nuclear division has already taken place. Here also you can see one cell with two nuclei, one cell with two nuclei. Okay, here. And in this figure, you can also see cell division. There are two different cells, sibling cells. So this is the entire process of mitosis. One by one, we are going to see with all the steps. Let us start with prophase. During this phase, chromatin condenses to form chromosomes. Here what will happen? As I said earlier, that chromatin network will condense and it will form chromosome. Condensation will take place. It will start over here. There are two identical copies of each chromosome which are attached to centromere. If you have studied the structure of chromosome, here you can see. In the center, there will be centromere. We have already done in 8th chapter. In all these figures, you can see. Here also you can see. It's not very clear, but still you can understand. Here at the center, there will be centromere. There are two identical copies of each chromosome which are attached to centromere. During the end of prophase, nucleolus will dissolve. If you have studied 8th chapter, you know what is a nucleolus. It is a body which is present inside the nucleus. Slowly and slowly, this nucleolus will get dissolved. Nuclear membrane will also disintegrate, and the centrosomes will move to opposite poles. Here, these are the centrosomes. Centrosomes with centriole pairs. You can see centrioles are arranged at 90 degrees. So they will start moving to opposite poles. Here they are moving to opposite poles. Spindle fibers will start to appear. This also you can see clearly. Spindle fibers have already begun to appear. So this is prophase. Second is metaphase. What happens in metaphase? Chromosomes will get aligned at the center at equatorial plate. Here, this entire line is called as equatorial plate or metaphasic plate. All the chromosomes will align here. Now, when you see this entire thing, this diagram or this phase is the best phase to study the structure of chromosomes. There are 4 types of chromosomes we have already studied. These chromosomes are divided into 7 groups. So, if a geneticist or a person who is working with chromosomes, who wants to do research on chromosomes, a cytogeneticist, when these people want to study about the structure of chromosomes, they study at this stage, at metaphasic stage. Why? Because the chromosomes are clearly visible. In prophase, they will be in a phase of getting condensed, so the structure is not very clear. In anaphase, they have already split it. In telophase, they are in chromatin network form. So this is the actual stage or the best stage to study the structure of chromosomes. So here it is written, the analysis of metaphase chromosome is used in cytogenetics and cancer studies. Third is the anaphase. This is the shortest stage of the cell cycle. What happens in this phase? The replicated chromosomes move apart and daughter chromatids move to the opposite poles. Here you can see this was first in metaphase, this was one chromosome. Now the sister chromatids have been split and they are moving to opposite poles. Chromosomes are maximally condensed during the late anaphase and the chromosomes appear Y-shaped as they move to opposite poles. Again, you can see this diagram. They appear as Y-shaped, right? Y or V shape when they are moving to opposite poles. And at the end, there is telophase. This is where the two daughter nuclei are formed. Again, nucleolus and nuclear membrane will reappear. You can see it here. Slowly and slowly, the nuclear membrane will reappear and nucleolus will also start to form. So this is actually opposite of prophase. This is the last phase of mitosis, and finally the chromosomes will reach the poles. And in one cell, 2 nuclei will be formed. So here we complete with karyokinesis. Now what happens after karyokinesis? The division of cell will start. And when the cell divides, it is called as cytokinesis. The division of cytoplasm, organelles and cell membrane to form 2 cells is known as cytokinesis. So here the cytoplasm will divide, the organelles will divide. The organelles have also become double in amount. So they will divide equally, as well as the cell membrane will also divide, and then 2 new cells will be formed. These 2 new cells are called as daughter cells. And these daughter cells in mitosis are exactly same as the parent cell or identical to the parent cell. Now let us see what is the importance of this mitosis. Mitosis produces genetically identical daughter cells. As we have seen earlier, that in mitosis the daughter cells are completely same to the parent cell. The chromosome number is also same and the DNA is also same. So they are genetically identical to the parent cell. Growth of the body occur via mitosis. Mitosis is the basic cell division of somatic cell. And because of mitosis, the cell divides, and because of cell division, the growth of the body occurs. If there is any problem in some cell, if there is any damage in a cell, then cell repair and replacement also occurs via mitosis. Another importance of mitosis is it is important in maintaining the nucleocytoplasmic ratio. What is nucleocytoplasmic ratio? It is a ratio of the nucleoplasm to the cytoplasm. Nucleoplasm is the fluid that is present inside the nucleus. So the amount of nucleoplasm and cytoplasm should remain constant. The ratios should always remain constant. Only then the cell will function properly. So the maintenance of this nucleocytoplasmic ratio is the duty of mitosis. That is the importance of mitosis. Now this was all about cell division and mitosis. Sometimes what happens, there are some cells which do not undergo mitosis. So actually we can say that they have exited the cell cycle. Some cells divide at a fast rate, whereas some divide slowly, and some even do not divide once they are formed. Those cells which do not divide once formed enter into a phase which is known as G0 phase. It can also be called as quiescent phase. They will not undergo cell division. They have exited the cell division, they have come out of the cell cycle. Example of this is neuronal cells or the nerve cells. What happens once they are formed, they do not divide. Even our RBC, once they are formed and matured, they do not divide. They complete their life cycle and then they die and new RBCs will be formed. Same is for nerve cells also. So we say that they are in G0 phase. I'll show you it in the diagram. See here. In the beginning, I've explained this entire diagram. Here you can see that some cells come out of the cell cycle and they undergo cell cycle arrest. So this is known as G0 phase. You have already understood G1 as G2 and M. This is the G0 phase, that is arrest of cell cycle or arrest of cell division. Now the cell will not undergo any division. So by this we complete our first part of this chapter, that is mitosis. In the next video, we are going to study another cell division, which is meiosis. Meiosis is a bit complex than mitosis. So before going to that thing, I want you all to study this very clearly. And one thing that you have to remember here is, instead of studying the points, instead of studying the theoretical part, I want you all to focus on the diagrams. If you're clear with your diagrams, if you know what diagram you need to make in prophase, what diagram you're supposed to make in metaphase, anaphase, telophase, then you will be able to remember all the points very clearly. So please focus on the diagrams. So let's meet in the next video. Till then, take care, have a nice day, and if you have any doubts, then you can contact me. But please keep studying. Thank you.